So last time we talked about the NMR instrument briefly. This is a schematic diagram of the NMR spectrophotometer, and you can see the different components. So here we have the magnet that we have been talking about, that you provide a magnetic field to your sample. This is your sample tube in which your sample is present in the form of a solution, and here you can find these blue wires. These are the radio frequency transmitter lines which provide radio frequency to your sample, and then it takes the signal to the computer where it is recorded in the form of a spectrum, right? So the classical instruments or the obsolete instruments that were used decades ago, they were known as the continuous wave NMR spectrophotometers, and this is what they look like. And we have discussed the basic principle of how, or the basic operation of how these instruments work in the previous session. So what we do in a classical NMR machine is that we scan from low field to high field. As we discussed previously, that you have to scan from a low field to high field, and as you increase the strength of the magnetic field, the Larmor frequency increases. But what we have done is that we keep the radio frequency transmitter at a fixed frequency. So when you keep it at a fixed frequency, it means that you have to take your protons, which are the radio frequency receivers in this case, you have to take them to a certain level, to exactly the same value of the frequency at which your radio frequency transmitter is radiating. You start increasing the magnetic field, and you see that the Larmor frequency of the protons, they start increasing, and what happens is that these protons, these different types of protons which are present in your molecule, they start resonating one at a time. As soon as their frequencies, their Larmor frequencies match the frequency of the radio frequency transmitter, resonance takes place and you see a signal. So basically, the basic operation of this machine is that you scan it from a low field to a high field, and we discussed it last time with the help of some calculations, how the radio frequency and the Larmor frequency, they get matched, and then you can differentiate between shielded and de-shielded, or more shielded and less shielded protons, right? So you provide a constant beam of radio frequency which is at a fixed frequency. Now when you do this, as I told you earlier, all of these different types of protons, they resonate one at a time. So it takes a lot of time. So it's a time consuming method, and it takes several minutes to plot one NMR spectrum. You might not have the luxury of spending more time on scanning your molecule again and again. So what happens as a result is that you have low number of scans. With low number of scans, you might have a high noise level. So your signal to noise ratio will not be very good. जब आप बार-बार स्कैन करते हैं, when you scan your molecule many times and you stack all these spectra together, the signals or the peaks from your molecule, they get enhanced with every scan, but the noise is cancelled because the noise is random, and the signals, they appear at the same frequency or at the same position. It's a slow process, and you might have a high noise level, which means that the signal to noise ratio will not be very good. So then scientists thought about another method of analyzing these molecules through NMR, and they came up with some modern instruments which are operated on a pulsed Fourier transform technology. So this is how it looks like. This is the NMR machine that is present in your institute right here. That is a 400 megahertz NMR spectrophotometer manufactured by Varian. So this cylinder that you see here, it has the magnet inside, the magnetic field inside, and you can see this inlet from where you can insert your sample, your sample in an NMR tube, in a sample tube. So when you insert it, it goes inside in the magnetic field, and then magnetic field is provided along with the radio frequency transmitter, which is also attached to this machine, right? And here you see some other inlets. These are for the liquid helium and the liquid nitrogen. Liquid helium is used to keep your magnet at a low temperature. So the magnet that is used inside this machine is a superconducting magnet, and liquid helium, you know, its boiling point is very low, so it keeps it very cool. That is in the inner core of this cylinder that you see. The outer core has liquid nitrogen. Liquid nitrogen again has a very low boiling point, and that is used to keep the liquid helium in liquid state. इसके साथ फिर ये console लगा होता है, takes the signals and it gives it to the computer, and then computer plots the spectrum. So these modern instruments, they are operated through modern softwares, and you can play with the software. You can change the pulse angles, you can change the pulse duration, you can have different information about the molecule when you switch from one technique to another, whether you want to see proton or carbon or nitrogen or any other information that you need. So the difference between classical continuous wave NMR spectrophotometer and a pulse Fourier transform NMR spectrophotometer is that in FT NMR, all protons are excited simultaneously. So we saw in the previous technique or instrument that all these different types of protons, they are excited one at a time. But in pulsed Fourier transform technology, they are excited simultaneously. This whole process, which involves the pulse, what is a pulse? A pulse means to turn on the radio frequency transmitter for a very short time and then turn it off. So what we do in this technology is that we turn on the radio frequency transmitter for a very short period of time and then we turn it off. So on off means that we are actually giving pulses of radio frequency to your molecule. This pulse excites all of the protons present, all of the nuclei present in your sample at the same time simultaneously. Then they are relaxed back and give you a signal in the form of FID. FID means free induction decay. So when you give pulse and you excite all the protons and then they are relaxed back, the signal is obtained in the form of an FID, free induction decay. It's a decaying signal, and you will see after a couple of slides that this signal decays with time. So it's a time domain signal. The signal that we obtained from the continuous wave NMR spectrophotometer, that was a frequency domain spectrum. But in this case, when you give pulse and you excite all the protons, the signal that you obtain as a result is in the form of FID, which is a time domain signal. And why it is known as FID? Because the signal decays with time. How it decays? Its intensity decreases with time. So it starts off with the maximum intensity and then it decays with time. So this is how it looks like. But this FID or the time domain signal, it's not of much use to us because we cannot interpret it. We cannot read it. The human brain cannot read it or interpret or extract information from this spectrum. So what we do is that we convert this time domain signal into a frequency domain signal by applying a mathematical operation which we call the Fourier transform, and that is why this technology is known as the pulsed Fourier transform technology. We convert this time domain signal into a frequency domain signal with the help of a mathematical operation which we call the Fourier transform. So the steps involved in pulsed Fourier transform technology is the pulse followed by acquisition of the FID and then conversion of the FID into a frequency domain signal through Fourier transform. So all of these steps involved in the process takes only a few seconds. While previously you saw that one spectrum could be obtained in several minutes. But here, within a few seconds, you can do the whole operation, and this is one scan. This whole process or all of these steps means one scan. So you scan your molecule once in a few seconds. So now you have enough time, you can scan it again and again. What that means is that if you can repeat this whole process again and again within a short period of time, it means you can enhance the signal intensity and you can reduce the noise levels. So when you reduce the noise levels and you increase the signal intensity, it will lead to a very good spectrum because the signal to noise ratio would be much better as compared to the previous technique or the continuous wave NMR technology. Now, what is pulse? Pulse, as I told you earlier, is turning on the radio frequency transmitter for a short time period and then switching it off. So one pulse means one scan. You give one pulse, then you allow the nuclei to relax back, and then you acquire the FID and convert it into frequency domain signal through Fourier transform. That is one scan. Right, so because of this pulse technology, you can obtain several NMR spectra within a very short period of time, and when you stack them together, the signal to noise ratio increases because the signal intensity increases and the noise is cancelled because the noise is random. So in the modern instruments, everything revolves around the pulse. Now this pulse can have different durations and different angles. That is very important, right? So this pulse or sequence of pulses is the heart of all NMR experiments. So you give different pulse sequence, you give pulses in different directions for different duration and different angles, and you get different information about the molecule. Now let's see what's inside the NMR machine. As I told you, that cylinder type structure, that is actually the having the magnet inside, and this is the inside of that cylindrical structure. So you see right in the center, this tube, this is the NMR sample tube. So you insert your sample in this tube with the help of a syringe. It's in solution form. You prepare a solution of your sample in a deuterated solvent. Once you have the solution, you insert it here. Here we have air pressure, and that air pressure allows it to be suspended on top here. And when you want to insert it inside this magnet, you just reduce the air pressure inside, and that is just by clicking a button. It reduces the pressure inside and then takes your sample tube inside the magnet. Right here, these green lines here, these are to indicate the NMR probe. There are different probes used in NMR machines, that is for different purposes. Right? If you want to have normal analysis under normal conditions of temperature at room temperature, right? So you have a normal probe. But if you want to analyze your sample at a low temperature, then you have to change this probe. And for that you can have some specialized probes like cryoprobe, Cryoprobes are the probes in which, with the help of which you can analyze your sample at low temperature. So you can change the probe accordingly, according to your sample, according to the requirements of your analysis. These dark green lines indicate the magnet shim assembly. Now what is that? You are providing a magnetic field of constant strength. You do not change the strength of the magnetic field during this experiment or during operation. But still, this magnetic field could be inhomogeneous. It may not be homogeneous. Because of several factors, your sample may be getting an inhomogeneous magnetic field. What we do is that we shim it. So shimming can be done with the help of certain commands that you give with the help of your software in modern instruments. So this is for shimming and providing a homogeneous magnetic field. Then you see these wires here, and you see that these wires are inside a container, and that is the container which has liquid helium inside. So these wires are for the magnet, which provide magnetic field. So this is your magnet, the superconducting magnet that you are using. So these are the coils of the magnet, and they are immersed inside liquid helium. This pink color shows that it is filled with liquid helium to keep these coils at low temperature, to keep it as a superconducting magnet. On the outer core, we have another container that contains liquid nitrogen. This liquid nitrogen is used to keep this liquid helium in liquid state. So because this is in direct contact with the external environment, so it's more obvious that this liquid nitrogen will evaporate more quickly as compared to the liquid helium, which is on the inner side. So we usually fill this liquid nitrogen every week, and you are required to refill the helium after 6 or 8 months. So this was all about the NMR machine.